Hello, dear students. In this series of 6 videos, we are going to talk about nucleic acids and protein synthesis. My dear students, this chapter of A levels biology, it has three major parts. Number one, we need to know about the nucleic acids like DNA and RNA, their structure. Then, for the second half, we need to talk about a very important experiment that was performed by Sir Messelson and Stahl to explain that DNA replication was semi conservative. And number three, we have the third half, which is about protein production and how proteins are being formed inside the body. So after each and every video, after first half video, we will move towards the P1 and P2 questions, and then we'll move to the Messelson and Stahl. Then again, so this is our pattern. Let's talk about it. Nucleic acids and protein synthesis. Right from the beginning, right from the start, scientists they were very curious in knowing that what is the actual molecule which is responsible for transfer of information from parents to offsprings. You might have noticed that people they often recognize you by looking just at your features, by looking just at how you walk, how you speak, how you the shape of your eyes, your hair, your fingers, anything. Any phenotypical or physical feature, they can know, they can estimate that you are जो है वो आप किसके बेटे हैं, आपके फादर कौन है और आपकी मदर कौन है. So this is because of this DNA. And right from the beginning, people were scientists very curious to know कि हमारे पास वो कौन सा ऐसा molecule है जो इसको transfer करता है. Initially, scientists and people, philosophers, they thought that female body acts like a land and male body acts like a seed. So when a male intercourse with a female, it introduces some sort of seeds in the body of a female, and as seeds germinate, इसी तरह babies जो है वो पैदा हो जाते हैं. And so on. There were many philosophies and theories about this. Later on, there was a lady, and her name was Rosalind Franklin. Based on her studies, two scientists, most celebrated and most known scientists, Watson and Crick, they gave this model of DNA. Watson and Crick. But initially, there was a thing which was in mind of each and every biologist, each and every scientist. Number one, anything which is responsible for this transfer of information, number one, it must be able to carry large information, and number two, it should perfectly copy and paste. एक ऐसा मॉलिक्यूल होना चाहिए नंबर वन जो बहुत सारी इंफॉर्मेशन को कैरी कर सके, एंड नंबर टू बिल्कुल परफेक्टली कॉपी हो सके और पेस्ट हो सके. वॉटसन और क्रिक ने जब डीएनए के मॉडल के बारे में बताया तो ये डीएनए का मॉडल इन तमाम चीजों के ऊपर 100% खरा यानी पूरा हो जाता है. सबसे पहले लेट्स टॉक अबाउट दिस डीएनए. डीएनए मींस डीऑक्सीराइबोन्यूक्लिक एसिड. Deoxy means it is something with one less oxygen. Ribo means it's a pentose sugar जिसमें हमारे पास 5 कार्बन होते हैं. And nucleic acid means it is something which has nuclear contents जो कि nucleus से related है. Now let's talk about this DNA structure. We know that DNA is a large polymer. And if DNA is a polymer, every polymer they have their monomers. डीएनए के मोनोमर्स न्यूक्लियोटाइड्स होते हैं. You might have studied in your O levels A, T, G, and C. Now, these A, T, G, and C, they are just one part of the nucleotide. First, let's talk about this whole structure. Whenever you see this DNA structure, this is a double helical structure, and I have made this in a straight form. ये बिल्कुल twisted नहीं बनाया हुआ. यहाँ पर हमारे पास दो रीजन्स ऐसे हैं. दिस इज दिस लार्ज लैडर, दिस लार्ज पार्ट ऑफ द लैडर, दिस इज अ शुगर फॉस्फेट बैकबोन. दिस इज अ शुगर फॉस्फेट बैकबोन. और इसी तरह यहां पर भी हमारे पास ये जो लार्ज आपको पार्ट नजर आ रहा है, दिस इज शुगर फॉस्फेट बैकबोन. अब हम बात करते हैं कि न्यूक्लियोटाइड का स्ट्रक्चर कैसा होता है और इसमें क्या क्या होता है. दिस न्यूक्लियोटाइड इज द बेस of DNA structure. DNA is a large polymer and its monomers are these nucleotides, and they contain three things. Number one, there is a ribose sugar. There is a ribose sugar, a 5 carbon sugar. This is carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5 is projected outside this whole ring. And here we have oxygen. Then we have a phosphate group. So when we say a sugar phosphate backbone, so this is sugar and this is phosphate, and they connect with each other to form a sugar phosphate backbone. And here we have an organic base. In your O levels, जब आप कहते थे ATGC, so adenine, thiamine, guanine and cytosine, ये चारों बेसिस कहलाती हैं. और आपने ये भी याद कर रखा हुआ है कि A and T they always pair together and G and C they always pair together. There are nucleotide bases and they always pair together. And let me tell you another thing, that while A and T they bind with each other, they connect with each other, there are two hydrogen bonds. इसके ऊपर बहुत सारे questions आने वाले. And while G and C connect with each other, they have three hydrogen bonds in between them. So A and T they have two hydrogen bonds, and G and C they have three hydrogen bonds. A always binds with T and G always binds with C no matter what happens. After this, so when we have a and a T bonded together, we call them a base pair. This is a base pair. We have a when we have a G and C bonded together, we have this a base pair. So this one is a singular base, this C is a single base, and when G and C are connected with each other, we call it a base pair. This is something which is also going to help you a lot in your exam. कि ये बेस पेयर है और ये सिंगल बेस है, ये भी आपको एग्जाम में बहुत हेल्प करने वाली है. Now, just like this, first let's talk about this structure. So for this nucleotide, we have a sugar here, we have a phosphate, and we have a base. Let's talk about the structure of this phosphate first. For exams point of view, you guys don't really need to remember these structures, but you need to recognize them because they may give you these structures and they ask you to recognize what are they. So this is a phosphate, this is a P, and then we have 4O, and we have a double bond here. So this is a phosphate. Then there are two types of sugars on your screen. We have a ribose and we have a deoxyribose. They have a similar structure with just one difference. So this is carbon 1, this is carbon 2, carbon 3, carbon 4, and this is carbon 5. For a deoxyribose sugar, on carbon number 2, we have one oxygen less. There is no OH there. We just have a single H. And for ribose, for RNA, we have 1, 2. On this carbon 2, we have an OH. So we have OH and OH here, and here we don't have this O. So that makes DNA की sugar go, this makes a deoxyribose. It's a ribose with one oxygen less. And for RNA, it's an oxyribose. It has an oxygen which is attached with this H at carbon 2. This is the first difference that you are studying in my class in the Slate studio in DNA and RNA. We are going to talk about more differences in upcoming class. All right, now we have purines and we have pyrimidines. So in order to remember pyrimidines, let me tell you one thing, cut and dine. आप इसको याद रखें, कट एंड डाइन, कट एंड डाइन, कोई भी तरीका जिससे आपको याद रहे. सो कट से क्या बनता है? कट से बनता है सी से साइटोसिन, यू से यूरासिल, आई टेल यू लेटर व्हाट इज दिस यूरासिल, एंड टी से थायमीन. रिमेंबर, कट एंड डाइन, कट एंड डाइन. सो डाइन से पायरिमिडीन बनती है. So cut, CUT, cytosine, uracil, and thiamine, they are pyrimidines. And remember, they have a single ring structure. They have a single ring structure. And another important thing you need to remember is that in RNA, T is replaced with uracil, U. Whenever you have an RNA, you cannot see a thymine over there. If T is there, either that structure is wrong or it's not an RNA. In RNA, we don't have T's, we have U's there. So, In RNA, T is replaced by a uracil. So I hope you will remember this cut and dine thing. Then for purines, you need to remember it like pure as gold. Pure as gold. So pure stands with the purine, as stands for the adenine, and G gold stands for the guanine. Pure as gold, they are purines. Cut and dine, they are cytosine, uracil, thiamine, and they are pyrimidines. So remember one thing, that purines, they always bind with pyrimidines. As you can see, A binds with T or U, and here G always binds with C. No matter it's a DNA or an RNA, G always binds with C, right? So now, my dear students, we need to talk about the differences. Let's talk about this DNA structure now. For this DNA structure, you can see one strand is lying in an opposite direction and the other strand is going in the right direction, right? So they are actually anti-parallel to each other. One is going like this and the other one is going like this. So this DNA is a very small molecule. It's a very small thing. We cannot see it with our naked eye or even with the electron microscope. Then how do we know that on which part of the DNA we are referring to? So, in order to refer to certain parts of DNA, in order to know about the area or a region of a DNA of which some scientists are talking or they're exchanging information, we have to keep some nishaniya. So, वो निशानियां क्या होती हैं? We talk in this way. Here, we know that this is carbon 1, this is carbon 1, this is carbon 2, this is carbon 3, this is carbon 4, and here, this carbon 5 is available for further bonding. Here in this structure, this is carbon 1, 2, 3, 4, and here carbon 5 is available for further bonding. So we call it a 5' end. We call it a 5' end. And similarly, like this is carbon 3, this is carbon 3, this is carbon 3, and this is carbon 3. Right? This 5 is engaged with another bonding. This 5 is engaged with another one. 5 is engaged with another one. So here, at the end of this strand, where this end was a 5' end, at the end of this strand, we have a strand which is a 3' end. So we can say this is 5' end of a DNA and this is 3' end. And on the opposite side, since we know that this strand is going in an anti-parallel direction, we have a 3' end over here and 5' end. So 5' end is facing a 3' end, and here 5' end is facing a 3' end. This is something of very prime importance that we are going to study in our past papers. So I thought, okay, why not tell you now? All right. Now, now we need to talk about RNA. My dear students, let me tell you one thing, that RNA is a copy of DNA. Let me tell you that RNA is actually a disposable copy of DNA. And we have three types of RNA in our bodies. Number one, we have an rRNA. It is called ribosomal RNA. Number two, we have a mRNA. It is called messenger RNA. And number three, we have a tRNA. It is called a transfer RNA. Actually, our DNA is a very precious information. And cell never ever wants to include that precious information every time when the cell wants to make a protein. And this might happen for thousands of times for a cell, for hundreds of times, for thousands of times for a cell, which is involved in protein production, like the cells which are involved in enzyme production, like the cells which are involved in hormone secretion or antibody production. So the original information DNA that is conserved, that information is a precious information that is not shared every time. What happens is that cell it directs this RNA to form from this DNA. It's just like a Xerox copy. ये बिल्कुल जिस तरह से आपकी फोटो कॉपी होती है डीएनए की, आप जैसे कि कोई बुक किसी को नहीं देना चाहते हैं तो आप उससे कहोगे कि मेरी बुक को कॉपी करवा लो, बिकॉज ऑल अबाउट व्हाट यू आर कंसर्न विद इज द इंफॉर्मेशन। हाँ, तो इंफॉर्मेशन आपको फोटो कॉपी से भी मिल जाएगी। ओके, तो होता क्या है? होता ही है बेटे कि हमारे पास डीएनए की जो ओरिजिनल इंफॉर्मेशन है, मैंने इसको डेलिबरेटली ऐसे बनाया हुआ है, डोंट जज मी। ये जो आपके पास डीएनए की इंफॉर्मेशन है, एक प्रोसेस होता है, जब हम इसको अभी हम डिटेल में पढ़ने वाले हैं। जब इस डीएनए से आरएनए बनता है तो जहां जहां टी था वहां वहां तो ए लगता है, बट जहां जहां ए होता है यहां टी आना चाहिए था, यहां यू लगता जाता है। सो अगर इस डीएनए को कॉपी करके आरएनए बनाया जा रहा है तो जहां जहां कॉम्प्लीमेंट्री टी ऐड होना था वहां वहां कॉम्प्लीमेंट्री यू ऐड होता है। सो नाउ यू नो अबाउट द टू डिफरेंसेस। नंबर वन, it is having a sugar which is different from the sugar of DNA. And number two, it is always going to have U instead of T. Now let's talk about a couple of more uh, differences between DNA and RNA. Do you remember this lady from Game of Thrones? She's mother of dragons. I got this meme from Google, someone sent me, so I thought why not to include it. She has this, एक तरफ इसके बाल जो है they are going single stranded और दूसरी तरफ जो इसके बाल जा रहे हैं they're going double stranded. सो आरएनए और डीएनए की पहला डिफरेंस जो कि आपको आई एम श्योर कभी भी नहीं भूलने वाला, वो ये है कि आरएनए इज अ सिंगल स्ट्रैंडेड मॉलिक्यूल like this, and DNA is a double stranded thing like this. Okay, let's talk about these differences. For RNA, it's a single polynucleotide chain, single chain, and DNA, it's a double helix. जो भी मैंने आपको ये मदर ऑफ ड्रैगन्स का दिखाया. Then RNA, it has a smaller molecular mass, and DNA, it has a larger molecular mass. Then RNA, their pentose sugar is ribose sugar. Their pentose sugar is ribose sugar. And for DNA, their pentose sugar is deoxyribose. They have a deoxyribose. For RNA, organic bases present are adenine, guanine, cytosine, and uracil. And there we have adenine, guanine, cytosine, and thymine. Here, ratio of adenine to uracil and ratio of cytosine to guanine, it always varies because it's a single-stranded structure. But here, in this DNA, adenine to thymine ratio is always same and guanine to cytosine ratio is always same. यानी अगर आपके पास ये एक स्ट्रक्चर है, एक तरफ जितने एडेनिन होंगे उस तरफ दूसरी तरफ उतने ही थायमिन होंगे, और एक तरफ जितने साइटोसिन होंगे दूसरी तरफ उतने ही गवानीन होंगे. So the ratio is same in DNA. Then, DNA is mostly found in nucleus, with some present in mitochondria and chloroplast. And RNA, it is manufactured in nucleus, but it is never found in nucleus. It is always found in the cytoplasm. Then, RNA amounts. This RNA amounts, it varies from cell to cell, right? And DNA amount, they are always constant. DNA की amount जो है वो vary नहीं कर रही हो. Then, chemically, RNA is less stable. As I have said that it's a disposable copy of a DNA, it's chemically less stable, but DNA is chemically very stable. Then, three basic forms में RNA मिलता है: messenger RNA, transfer RNA, and ribosomal RNA. And you're going to know about it when we are going to talk about protein production. So जब protein production पढ़ेंगे, then you will understand these types and roles of RNAs. But DNA, they have only one basic form, but with an almost infinite variety within that form. So this is the difference between DNA and RNA. So, Bachu, this was the basic information that you needed to know about DNA and RNA, and there are number of multiple choice questions which you can attempt by knowing such information. So, in our next video, we are going to talk about the past paper questions which are related to whatever we have studied here, and then we will talk about the semi-conservative replication on DNA. So, stay with us, stay with Slate, and share it with your friends as well. Bye bye.